Hi everyone, today I'm going to talk about how mitochondrial dysfunction regulates APOE expression and secretion. And this is work that's been funded in part by the ADRC at Emory. So to start out, I want to talk about a couple of models for how mitochondria are able to influence Alzheimer's processes. So in a more typical model, mitochondria are downstream from A beta plaques and the APOE4 allele. APOE4, of course, being the strongest genetic risk factor for late onset Alzheimer's disease. It's been shown that both plaques and the APOE4 allele lessen respiratory gene activity. And so in this way, mitochondria are just acting kind of in response to plaques and APOE4, and they're downstream. An alternative model is one where mitochondria are able to influence APOE and A beta plaques. And in this model, mitochondria would be part of a feed-forward loop that's contributing to disease processes. And so this is the model that we favor based on the data that I'll show you. And a prediction of this model is that genetic or pharmacological disruption of mitochondria would affect APOE expression. We'll be focusing on this link between APOE and mitochondria. And to test this hypothesis, we targeted complex 3 of the electron transport chain with antimycin in iPSC-derived neurons and astrocytes. And we did this with an ELISA-based assay. So if you look here, in gray is the vehicle condition, in blue is the antimycin treatment. For the neuronal lysates, we saw no change in APOE levels. And looking at astrocyte lysates, we saw about a 2-fold increase in APOE levels. And then in conditioned media from astrocytes, we saw a similar about 2-fold increase in APOE levels. And so we were able to look at the APOE that's secreted into the media over these 48 hours of antimycin treatment. And we looked also at the mRNA level at APOE levels for first here, the astrocytes, where again, in gray, we have the vehicle treatment, and then in blue here, we have the antimycin treatment, and we saw a significant increase. And this was done using nanostring, which is an mRNA quantification that doesn't use any amplification step. And we looked also at a couple of genetic mutant mitochondrial lines, one lacking the protein SLC25A1, which is a carrier that moves the metabolite citrate on the inner membrane, and then a second line, which was lacking the ATP translocator on the inner membrane. And when we look at both these cell lines, we see a very robust APOE upregulation. And this is just a loading control showing these different samples were loaded about evenly. Looking more at the results from this nanostring panel, which was an 800 gene panel looking at neuroinflammation. These are the volcano plots, where for both the cell lines we tested, you can see the yellow are the upregulated hits. We see APOE here, as I highlighted previously, and then the same is true in the antimycin treated astrocytes, where we see APOE is an upregulated hit. And up top here is the proportion of the panel that was a significant hit. So comparing the cell lines to the astrocytes were had more hits here. But regardless of the number of hits, when we look at the gene ontology terms that are related to the upregulated hits in both cell lines and the antimycin-treated astrocytes, we see inflammation popping out, inflammatory response, cytokine signaling, cytokine receptors. And so this suggests that the mechanism linking APOE and mitochondria could be inflammatory signaling, although we need to follow up on that. And this also shows that mitochondria are able to drive neuroinflammation. And so to sum up, I've shown you that inhibiting complex 3 of the transport chain is able to cause APOE upregulation, that this APOE upregulation co-occurs with increased inflammatory pathway signaling, and that mitochondria could act upstream in Alzheimer's disease through modulating APOE-dependent disease processes. So this is a way that mitochondria can influence Alzheimer's outside of just bioenergetics. And we think it would be interesting to look more into alternative ways that mitochondria can influence disease outside of just bioenergetics. And I do want to acknowledge my lab and our collaborators, the core facilities that helped us, and my funding sources, including the ADRC at Emory. And we're about to release this work as a preprint, so please check that out if you're interested. And thank you for your time.